we have Professor Barry Bavister. Barry was the first person ever to witness IVF in humans while working alongside Bob Edwards in 1968. He was co-author with Edwards and Steptoe of the first and second published papers describing IVF in humans, and also editor of the book The Mammalian Preimplantation Embryo. Barry was previously Professor of Conservation and Reproductive Biology at the University of New Orleans at the Audubon Center for Research of Endangered Species and part of the Audubon Nature Institute.
It's always an honor to talk about Bob Edwards. I was a high school dropout, and I left school at 16 and became a lab assistant for 3 years. I realized, however, that wasn't going to get me very far. So thanks to some good friends and Peterhouse College, Cambridge, I was able to get back into university and enroll as an undergraduate in 1964, 60 years ago. And I studied physiology primarily. It's become the love of my life. I taught it for many years. And after about 3 years of rather boring lectures, finally, a man came into my life who was dynamic and exciting, and that was Bob Edwards. And he lectured to us on reproductive physiology. What a fantastic lecturer he was. He was so enthusiastic about his topic that it just enthused me. However, his main focus, even at that time, was infertility, which was largely ascribed to blocked oviducts in women. I think male infertility wasn't recognized very well at the time, but certainly that was a major problem, causing infertility for up to 15% of couples. Surgical intervention to unblock the oviducts so the sperm and eggs could meet again was not very successful. So Bob, even in his lectures, had this brilliant idea that he might be able to use in vitro fertilization to overcome the problem. And I remember his exact words from 60 years ago. And he said, "Wouldn't it be wonderful if we could take an egg from a woman's ovary, fertilize it in vitro, and transfer the resulting embryo into a uterus so that she could have a normal, healthy baby?" Well, as I wrote later on, that was pure science fiction because no one had ever fertilized human eggs in vitro. There were a lot of problems with that. I was so enamored of Bob's lectures that once I finished my undergraduate degree in 1967, I rushed over to his little sub-department called the Marshall Laboratory and joined that lab as a PhD student, again, thanks to Peterhouse College. Now, Bob was not my PhD supervisor. He already had his hands full with Richard Gardner and Martin Johnson. But he did give me some advice because I asked him, "Bob, what do you think I should work on for my PhD thesis?" And he was a very canny Yorkshireman. And he said, "I think you should study in vitro fertilization." And I think, pretty sure that he hoped I might discover something that would help him because he had been trying to fertilize human eggs in vitro for several years with absolutely no success. And I think he was getting desperate. So I started my PhD research in 1967, and I needed an animal model. Well, the only available animal model for in vitro fertilization those days was the golden hamster. And that IVF had been reported in 1963 and 64 by a young Japanese scientist named Ryusho Yanagimachi working in the USA. What he did was to use a modified kind of a salt solution called pyrrole solution for in vitro fertilization. And so, of course, I just copied his method. And I'm very happy to discover that I could fertilize hamster eggs in vitro quite nicely, except it only worked occasionally. I would get highly erratic results. Sometimes it would work on a Monday, and I get 100% fertilization. And then I did the experiment again later in the week and got nothing. This went up and down for weeks. I had had the good fortune or luck, since I had been a lab assistant, to include a pH-sensitive dye in the solution called phenol red. And it turns yellow when the pH is acidic and purplish when it's alkaline. And so after about 6 or 8 months of work, I finally put it all together. So this is the result of about 6 or 8 months of work on my part, and fertilization percentage on the left, pH at the bottom. And as you can see in the bottom left-hand corner, when the pH is 7.2 or less, I get absolutely no results. And when the pH is as high as 7.8, almost all the eggs are fertilized, straight line relationship almost. And at pH 7.8, some of the eggs even have multiple sperm inside them. Well, I had immediately realized that fertilization was pH dependent. I was probably the first person to report that. And so I modified the thyroid solution from Yanagi-Machi by doubling the amount of sodium bicarbonate in the medium, which pushed the pH up to about 7.7 or so all the time. And then I also added an atmosphere of 5% carbon dioxide. So I had actually a bicarbonate CO2 pH buffer. And in doing so, I had reinvented the wheel, because that's the buffer system that we use in our bodies. But from that point on, using that modified medium, I could get fertilization almost 100% of the time. And I was now ready to move on with my PhD research. Well, I was now ready to move on with my PhD research with my new hamster fertilization medium, which Bob always called Barry's magic medium for some reason. But as soon as Bob heard about my result, he came rushing into the lab. He worked next door and said, Barry, why don't we try your magic medium with human eggs and sperm? And I think by that time he was desperate to try anything that might work. And so I said, certainly, certainly. So in the summer of 1968, we started working with my Barry's medium on human eggs and sperm. The way that this would work is that Bob would obtain eggs from bits of ovary that were donated by various clinical colleagues and prick out the immature eggs from the follicles and culture them for 36 hours so they could be fertilized. And that took until about the following evening at 6 p.m. So by that time, we were ready to inseminate the eggs. And so we did that for the first time. And I have to backtrack a little bit there, because years before, in 1951, my PhD supervisor, who was called Bunny Austin, who was the head of the lab, had discovered in rats with in vivo fertilization that spermatozoa could not immediately fertilize eggs when released from the male tract. They had to incubate for a while. And he called this process capacitation. Nobody knows what it was, still don't know, but we had to pre-incubate the sperm for a while. We didn't know how long that would be in humans. So after mixing the sperm and eggs together about 6 p.m., Bob said, let's come back in a couple of hours and see if we have fertilization. So we did that. We came back to his lab about 8 p.m. that night, and Bob eagerly sat down at the microscope and took out the first egg from the incubator and put it on a slide and looked at it under high power microscopy. He was so eager, I could see the expression on his face right now. But there was nothing. He couldn't see a sperm in the egg. Very disappointing. He called me over, Barry, take a look. No, there's nothing there. So we gave it another half hour, and Bob took the second egg out of the incubator, put it under the microscope, all eager, must work this time, but no, nothing there. And he called me over, and I'm sorry, Bob, it's not working. At this point, Bob was dejected. Human IVF was not going to work, and certainly my medium wasn't going to do the trick. And he's exhausted his repertoire of ideas for making human IVF work. So there, Bob is sitting in his chair, slumped against the wall, looking really dejected. Bob said, Barry, you look at the next one. He'd given up. It's over. So I sat down at the microscope chair for that time. And this picture shows the tiny, tiny human sperm inside a human egg. This is the first picture of an egg starting to undergo fertilization. The top arrow shows a black bar, and that's the mitochondria, the powerhouse of the sperm. There's a faint sperm tail at the back on the left. You can't see it in this picture, but it certainly was there. But most importantly, the bottom arrow shows an expanding sperm nucleus, and that's going to become a pronucleus, which I'll show you in a moment. And that said, this image was our eureka moment in 1968. And I remember exactly what happened almost 60 years ago now. I sat back, turned to Bob, and said, Bob, there it is. And he suddenly became alive. He rushed over to the microscope, sat down, saw the same image you're looking at, minus the arrows, of course, and then turned to me and said, Barry, we've done it. And that's exactly how he sounded. We looked at the rest of the eggs, half a dozen or so, and found that several of them, not all, showed similar signs of penetration of the egg, which is the first the start of in vitro fertilization. Fertilization takes about 18 hours total. It's a series of events, but the sperm in the egg is the very first step. We did numerous experiments over the next few weeks, more advanced stages of fertilization. And so here's a low power picture of an egg. And in the center are two circles, they're actually spheres. And those are the male and female pronuclei containing the respective DNA. When those two fuse, fertilization is complete, a process called syngamy. And that takes about 18 hours after sperm penetration. Based on all of these results over several weeks, Bob wrote two papers. The one that's less well known, he gave me first authorship, and that was Bavister, Edwards, and Steptoe. Now, he included Patrick Steptoe, his medical colleague, in that, even though Patrick wasn't involved in this bit of research, because he wanted very much to cement the relationship with Patrick, who was at the time trying to recover in vivo matured eggs that could eventually perhaps produce a baby. And then, of course, the major paper that's been referred to already by Jenny, I think, was Edwards, Bavister, and Steptoe. The publication date announcing the first in vitro fertilization was February 15th, 1969, but it appeared on Valentine's Day, the day before. And the result of having announced the first human IVF and being Valentine's Day caused an absolute explosion of interest from the press. Press journalists came from all over. Not all of the response, as Jenny pointed out earlier, was positive. There was a fair amount of negative response, the worst of which was that some, shall we say, journalists said that this IVF business would lead to the production of hordes of Nazi stormtroopers in the lab. I don't know whether they're all supposed to fit in the lab, but that's what they said. It was really nonsense. Bob, of course, at that point went on with his work with Steptoe and his colleague, his partner. She was not his assistant. He insisted on that. Jean Purdy, who was a friend of mine, and she was the world's first embryologist. And they worked up in Oldham to fertilize human eggs in vitro. I should just say that at that point, I'd had enough and I had to get back to my PhD research. I could not find an academic job in Cambridge, so I went to the United States and worked in Yanagi Machi's lab, which was a big plus for me. But Bob and his team worked steadily for about the next 9 years. Now, they could easily fertilize eggs in vitro that had been matured in vivo, recovered from volunteer patients. They could make lots of embryos, but after they transferred them, they got nothing. They tried this several hundred times and did not have a single uterine pregnancy. In desperation, I think, they said, let's stop hormonally stimulating the patients to produce lots of eggs. Let's just do natural maturation. And that was the process without any stimulation that produced the embryo that became Louise Brown. So this is the famous slide that started it all, and of course the press furore is intense after this. But here, Edwards and his team had proved point of principle. And as he had said years earlier in my class, this method, this IVF business, could produce a normal, healthy baby. He proved that. And at this point, Bob went on to become the focus of IVF and also the lightning rod for its criticism. And he continued with that for almost the rest of his life. I asked Bob when I saw him in 2007, you've done so much good work. Why haven't you received the Nobel Prize? And he said, well, I've been nominated 19 times, but it hasn't happened yet. Well, it took 3 more rounds of nomination until 2010, when he did receive the Nobel Prize in physiology or medicine, and it was obviously richly deserved. The Nobel Committee in 2010 put out a website where people could write tributes to Bob, and there were thousands of them, and I read a few of them. And then I stopped when I read this one. And this tribute to Bob Edwards obviously came from a man who had been an IVF baby, and his tribute was very simple. He said, "Thank you for my life." I get goosebumps every time I read that. I think those 5 words perfectly summarize Bob's career and his scientific efforts as a great humanitarian and a great scientist. And I think it's a wonderful epitaph for an incredible scientist. And it was my privilege to work with him for just a few weeks in 1968. That was a very brief thumbnail sketch of how we discovered human IVF. And if you would like a more detailed account, I wrote an article. It's not published, but it's called The Beginning of Human IVF. I can send you a PDF format free. Just write to me at barrybavister@gmail.com, and you can read a bit more detail about how the first human eggs were fertilized.